The thylacine, the Baiji, Spixes macaw. What do all these species have in common? Well, besides the fact that they're all very cool and awesome species that have existed, all of these are species that have gone extinct, partially due to human interference. Yeah, go on, feel bad about the actions of our species. We all deserve that guilt. While we're out here thinking about how bad you feel about the deaths of these species, and the so many others that have been erased from this plane of existence, let me tell you about a little concept we the people like to call de-extinction. That's right, no longer are your Jurassic Park dreams a thing of fiction, we now have the means to resurrect these long-lost species, regardless of the consequences. Okay, by now you may be excited about the possibility of bringing the dead back to life. And I am sorry to let you know that no, that's not what this is going to be all about. I know. But Elena, why would you tell us about all of this when that's not what you're going to be telling us about in the first place? Well, these subjects do happen to be related, because guess what? Instead of using the technologies we would use for the de-extinction process for, well, de-extinction, we can use it to stop species from becoming extinct in the first place. Let's get into it, shall we? So before we really get into this, I should probably answer the question of what is de-extinction and how does it work? Well, de-extinction, to put it quite simply, is the process of bringing an extinct species back to life. Pretty straightforward, right? It's a pretty fine concept, but how do we go about unextincting? De-extinction grammar is a little weird, pardon me. How do we go about bringing these species back to life? Well, here's where we branch out a bit. One of the more straightforward ways, well, not really straightforward, but one of the more predictable ways to go about bringing the dead and gone back to life would be through the use of gene editing, and to that extent, cloning. Specifically, nuclear transfer and germ cell transfer. Nuclear transfer, put sort of simply, involves taking the nucleus of a cell with the full set of genetic information and then implanting that nucleus into a nucleus-less egg cell so that it develops an embryo and can be birthed by a surrogate mother. Okay, it's not that simple, but if it works, the newly birthed individual should be a clone of the individual whose DNA/nucleus was used. Textbook cloning. This method is one that has been in the forefront of de-extinction efforts recently, having been successful in cloning species such as the gastric brooding frogs, otherwise known as platypus frogs, and Przewalski's horse. Germ cell cloning is similar, but it instead involves the editing and transfer of the primordial germ cells of the embryo. Primordial germ cells are the cells that develop into the various gametes, cells used in the process of reproduction, typically sperm or egg cells. By editing the germ cells of an individual, it is possible to directly affect the traits that their offspring will have. Germ cell transfer is useful in scenarios where nuclear transfer isn't necessarily viable, such as in the case of birds, because, birds lay eggs, and the way that those eggs are formed doesn't really leave room for nuclear transfer to be an option. And that pretty much sums up the gene editing and cloning part of de-extinction. Selective breeding for traits is also a method used in de-extinction, but there may be some debate as to whether or not this can be considered truly bringing a species back to life, as the genes that form the desired traits may be quite different from those of the species that is being worked to bring back to life. In just about all organisms, the genes coded in their DNA correspond to different proteins, which are sort of formed through the processes of transcription and translation. With selective breeding, the genes and proteins that they code for would most likely differ between the extinct species and the attempt to bring them back, which, as I've already stated, could call into doubt some of the validity, of the species truly becoming unextinct. Progress has already been made in the de-extinction efforts to date, such as the successful cloning of species such as the bucuro and, as mentioned before, the platypus frogs. Although successful is a relative term, while the de-extincted individuals did have the genetic material of their respective species and were technically alive, none survived more than a couple of days. Even more recently, a cloned Przewalski's horse, or P-horse as they are sometimes referred to, was born using frozen DNA from the San Diego's Frozen Zoo. More on that later. Of course, since de-extinction involves basically using science to defy the course of nature, there are some setbacks. These include the various factors such as the difficulty of actually finding and stringing together the DNA for certain extinct species, and finding suitable habitats for the recently unextinct species, but in the vein of the use of de-extinction for conservation purposes, a more relevant worry has to do with the possible lack of biodiversity that comes with a cloned/genetically engineered population. The genetic diversity of a population can act as a failsafe against certain less than optimal circumstances. In populations with a high level of genetic diversity, it's more likely that in the case of something like, say, a disease outbreak, there is a higher chance for the population's survival. While some more vulnerable species do usually have a low level of genetic diversity, it's not necessarily an indicator for the population being in danger. Like, it may surprise you to know that the primate with the lowest level of genetic diversity is actually humans, and we don't seem to have died out or gotten that close to it. There's also a factor of finding surrogate parents for the individuals created through the use of de-extinction technologies. Obviously, we can't grow these guys in some test tube in a lab somewhere, so they need to be incubated in another actual living body acting as the surrogate. So we would end up typically using a surrogate from the closest still living relative to the species that we are essentially resurrecting. But what if it just isn't possible for us to do that? What if even the closest relatives are incompatible with the birthing needs of the species? In order to successfully use de-extinction processes such as cloning, we need to know how to circumvent these obstacles. Okay, now that we're somewhat familiar with how de-extinction would work, allow me to introduce our featured species. So I'm just going to show you all a clip from a video here. This is from a video posted by the BBC, British Broadcasting Corporation, and it features an interesting individual of our featured species.
Huge. It's actually nice just seeing him wandering around in the forest undergrowth.
You can see he's got very good camouflage.
He has. That colour is incredible.
So, evolution hasn't entirely rendered him useless.
Okay, context time. This clip is part of a video taken from the BBC series Last Chance to See, which aired in the United Kingdom from September to October in 2009. This clip is from the 5th episode of the series and is posted on the BBC YouTube channel with the title Shagged by a Rare Parrot. Yeah. The episode is titled after the species of bird that is featured here, the kakapo. The kakapo, also known as the owl parrot, is a species of parrot that is native to the islands of New Zealand. The kakapo used to be relatively abundant in New Zealand until about the 1800s, when European settlers brought in new predators such as rats, stoats, and cats, which caused a sharp decline in the kakapo population. The population reached an all-time low of 18 individuals in 1977, which is quite recently if you think about it. But thanks to conservation efforts, the population has been on an upward trend. Currently, the kakapo population consists of about 208 individuals, which has earned them the category of critically endangered on the IUCN Red List. It's not really much of an accomplishment. So, how would we go about using de-extinction to help the kakapo? Well, first of all, in order to talk about what we can do, we should talk about what has been done. Since the 1950s, the government of New Zealand has made an effort to grow and preserve the population of kakapo that are still here today. Currently, the kakapo population is limited to 6 predator-free islands in the New Zealand archipelago, with the individuals that inhabit the islands being monitored closely. The reproductive practices of the kakapo population are also closely monitored and, I don't want to say manipulated, but honestly they kind of are. With a population that small, inbreeding is a real risk for the kakapo population, especially since the result is typically unsuccessful hatchings, which sucks, as kakapos already mate and reproduce so infrequently. Typically mating happens every few years or so. So the solution is using artificial insemination to basically make sure that the offspring of any given bird has as little chance of inbredness as possible. Seriously, they'll even use drones to override mating between individuals who are, quote-unquote, incompatible. It's a little weird, but all in the name of conservation, right? So, all that's fine and dandy, but where could we fit de-extinction technology to help with this? Well, artificial insemination requires doing the inseminating. Oh, that's gross to say. Anyways, in order to do the thing, they need to collect the material used for insemination from members of the kakapo population. I mean, without the actual genetic material, How are you supposed to make the child? So, while we're in that vein of discussion, let me bring your attention back to the video clip I just showed you. Featured in this clip is a particular Kakapo named Sirocco. Sirocco is an interesting case among the Kakapo, particularly for his preference for the company of humans, especially intimate company. This particular quirk of Sirocco's makes it particularly hard for those involved with Kakapo conservation efforts to collect his genetic material for artificial insemination. And that kind of sucks because without his traits in the population, there's less diversity. But what if we could use de-extinction technology, like germ cell transfer and such, to bring Sirocco's genetics into the population without using the typical genetic material that would be collected from him? Using germ cell transfer to create a clone of a bird in question needed for reproduction, or at least one carrying some of their genetic material, would allow those traits to be introduced into the population, even if the bird is not actively participating in the reproduction process. And that's cool, but we could also take it a step further. With germ cell transfer, we could potentially alter and add certain traits to the Kakapo offspring to increase the chances of successful reproduction and the survival of offspring. Certain traits could be changed to, say, alter the reproductive cycle so that the mating cycle is less infrequent, or make sure that most of the offspring actually can make it out of the egg and survive into adulthood. But wait, why would we use something made for bringing extinct animals back to life to keep them alive? Isn't that not what it's supposed to be meant for? Is what you may be asking now. Well, before you get your britches in a twist over de-extinction being used in cases pre-extinction, allow me to introduce you to the Przewalski horse. These horses are the last technically wild horse species alive today, as their ancestors were never really domesticated at all, and they inhabit a small region in Mongolia. Pea horses were once critically endangered and even went extinct in the wild, but thanks to conservation efforts, they have since returned to their habitat and are no longer on the brink of extinction. But that's not really the information that you're looking for, so let me introduce you to Kurt! Kurt was born in August of 2020. He was, created using the cloned genetic material of a Przewalski's horse kept in the Frozen Zoo, a database of genetic material kept by the San Diego Zoo. They did actually do this to make sure that that genetic material would be introduced into the population and further upping the genetic diversity of the Przewalski's horse population. And as I am not currently aware of any complications with Kurt's health, this is, to my knowledge, a success in the terms of cloning for conservation. That's fun to say. Back in 1990, when Michael Crichton's Jurassic Park was first published, the idea of using the DNA of extinct species to bring them back to life seemed like a mere fantasy, something that you would find in a science fiction novel, which, through Jurassic Park, people did. But since then, advances in the science of de-extinction has driven the development of many different technologies and practices that, while also useful in the actual de-extinction process, can also be used to aid conservation efforts for species that are in need today. And while we probably cannot, and most definitely should not, bring dinosaurs back to life as portrayed in Jurassic Park and other media, we can use de-extinction to make sure we never have to try to bring back their descendants. Thank you.